So I'm extremely honored today to introduce our distinguished speaker, Professor Garcia from University of Arizona. Dr. Garcia is truly an internationally recognized leader in pulmonary and critical care medicine, one of the most respected physician scientists in the field. He is an endowed professor in leadership and innovation at the University of Arizona Healthcare, an elected member of the National Academy of Medicine. He's a leader in the pathological studies of inflammatory lung injury and disease. He's also internationally recognized for examining the genetic basis of critical illnesses, such as ARDS, while addressing health disparities in vulnerable populations for the development of novel biomarkers and therapeutics for the critical ill, such as ARDS patients. He has published more than 550 peer-reviewed high-impact publications in prestigious titles, such as New England Journal of Medicine, Nature, AGR-CMB, the Blue Journal, the Red Journal, CHEST, etc. He has been extremely well funded by NIH, including 2 continuously supported PPJ grants, the program project grants, and several other ongoing R1 grants. He's also been well supported for this novel development of antibody drugs for the treatment of ARDS patients in this novel adventure of the biotech company he has established. Professor Garcia, besides being a renowned scholar and educator, is also a remarkable healthcare administrator. He was my chair when I was chairman of medicine, when I was assistant professor at the University of Chicago. So I know exactly how wonderful leader he is in nurturing junior faculties to make sure they develop and grow successfully and productively. And before that, he was the pulmonary division chief at Johns Hopkins. And after Chicago, he was the vice chancellor for research and vice president for health affairs at the University of Illinois, and later serving as the senior vice president for health sciences at the University of Arizona, where he oversaw a 1.2 billion health sciences and clinical enterprise. Since 2017, he has served as the CEO and founder of the biotech firm I just mentioned in developing this very remarkable antibody therapy for ARDS patients, for which no medicine has been available for the past 20 years. And Professor Garcia has received numerous awards, including the Edward Trudeau Medal, which is the highest honor given by the American Thoracic Society in recognition of research, mentoring accomplishments, and promotion of academic diversity. He's also very passionate in training physician scientists, other young scientists, and even high school students and with minority background. So he's very a true advocate for the young scientists for their development. Many of the students has ended up joining MD or PhD programs. So we're extremely honored to have Professor Garcia here today with us. Thank you so much for joining us and welcome to the CTSI seminar series.
Well, thank you so much, Linda. I very much appreciate that way over the top introduction. And let me just make sure I get my screen here going. Are we in business there, Elizabeth?
Yes.
Okay, cool. Well, it's an honor to be here and be part of this very renowned seminar series. I appreciate Linda and Steve Dubonet's invitation to participate in the series. UCLA is a great place, and I've already had several really interesting meetings through Zoom, and appreciate the folks on the Zoom here for participating in this particular lecture here. So you can see the title of my talk, Systems of Biology Enabled Journey to Dampen Acute Lung Inflammation. And of course, I have to disclose to you what Linda mentioned is that I'm a CEO of a company. I spun out a company called Aqua Lung Therapeutics. I'm the CEO and founder, and the main asset for this company is something I'll be talking about during the course of this lecture. And the understatement of the year, of course, is that this COVID-19 pandemic has impacted critical care illnesses in ways that are, are far from adequately measured to this stage, and will take quite some time to fully appreciate the impact of this virus on healthcare costs in this country and on outcomes. But if you look at, as you see here on the slide, if you look at just pre-COVID-19 estimates, critical care illnesses are a major consumption of healthcare costs. And in the thematic underpinning for the talk I'm about to share with you is the following, is that in critical care illnesses, a major driver of the outcomes in these patients is the development of multi-organ failure. And the development of multi-organ failure is directly linked to the activation of evolutionarily conserved inflammatory networks. Now, to have therapeutics that can address in a safe and efficient and hopefully targeted way the activation of these networks, and hopefully to dampen them or to block them completely is really truly a challenge. But unfortunately, we have not met that challenge in the critical care arena. And so unlike oncology and cardiology, where breakthrough drugs have emerged, the statins, Herceptin, et cetera, every drug currently used in the ICU for care of these critically ill patients is a generic one. So let's talk about ARDS. This is something that everyone on the Zoom, I'm sure, is very, very familiar with. I'm hoping Dr. Belperio is on the call as well. So I took a headline from his hometown newspaper, the Philadelphia Inquirer, here that talks about COVID-19 and the fact that a primary mechanism by which patients with COVID-19 infection succumb to that infection is through the development of ARDS. So this is there's a number of inflammatory stimuli that can produce ARDS, such as sepsis and trauma, bacterial pneumonias, as well as influenza pneumonias, et cetera. And before COVID-19, there was about 500,000 folks in the US every year that developed ARDS, 2 million people globally. And those numbers, of course, have been shattered with the onset of the COVID-19 pandemic. Before COVID, the mortality remained a staggering 30 to 40%. That mortality rate has climbed again within the COVID landscape as well. So what's the pathophysiology that leads to multi-organ failure and loss of life with ARDS? Well, the fundamental pathology is an increase in vascular leak due to inflammation, due to a variety of factors, but primarily inflammation that causes increased permeability. And this leads to leakage of fluid into the alveolar space. That makes the oxygen levels plummet. That makes the work of breathing enormous. And patients fatigue and require mechanical ventilation in order to survive. So looking at the two x-rays, obviously alveolar flooding is very, very prominent in the one on the right. And these patients need a ventilator and supportive care right now in low tidal volume ventilation, so that the ventilation parameters are not excessive, as well as prone ventilation has been used effectively, but they're very much supportive therapies. And an important point that I need to make, and I'll embellish later, is that although mechanical ventilation is a life-saving therapeutic intervention, because patients have experienced respiratory fatigue and they need a ventilator, the ventilator itself helps drive ARDS mortality. And we'll go into mechanisms on how that happens. So the bottom line is that the ARDS is an unbelievably significant and important unmet need. And there are no FDA approved drugs for treating ARDS at the present time. So to take you on the story about how we came across the role of the ventilator in ARDS and contributing to ARDS, this is a paper from many years ago, but it was when I was chief of pulmonary at Johns Hopkins. This is a study led by one of my faculty members, Roy Brower, who had the idea that the ventilator, in fact, based upon some good preclinical data, that the ventilator may be contributing to the bad outcomes in ARDS. And I was the chair of the protocol review committee for this particular trial. And while I was not excited about the nature of the trial, doing a trial where you just compare one tidal volume versus another seemed to be, not such an exciting clinical trial. As it turns out, this is the most impactful trial that the ARDS network ever did, because it's the only one that's produced any positive outcome. And what they showed here in this trial, Roy and his colleagues, they showed that if you compare a conventional tidal volume of about 12 mils per kilogram, this is the volume of the breath delivered to the patient every time the machine delivers a breath, and compare it to a lower tidal volume of 6 mils per kilogram, there was a significant benefit in survival. They measured biomarkers at the time of interleukin 6, and the blood was markedly reduced, significantly reduced compared to the conventional tidal volume. So the question that became, what's the mechanism by which the ventilator contributes to this this increased mortality? Obviously, increased inflammation, and so the mechanism for that has been pursued, and there's a lot of literature identifying contributory pathways and mechanical forces that contribute to that. But they all really relate to this, to the picture that I'm showing you, which is that this is a CT scan of a patient with ARDS. And it highlights the fact that ARDS, even though the chest X-ray looks fairly homogeneous, in fact, the lungs in ARDS patients are quite heterogeneously affected. So you can see there's an area there that's black that says overextension. You deliver a breath, that breath will overextend that area of the lung. You see there's areas of the lung that are flooded and collapsed. Those areas are not well ventilated at all. And that contributes to the shunt of blood through those areas, so that blood goes through and does not get oxygenated. And this is a very big contributor to the hypoxemia that you see in ARDS patients. But in the middle, when you have some alveolar units that collapse and then open with the breath, collapse and then open with the breath, this mechanical stress is thought to be a very important contributor to the injury, mechanical stress, and in fact, the inflammatory pathways that are activated. So at the time, we were very interested in developing some preclinical models that allowed us to identify what potential genes and proteins might be contributing to this process, might be potential biomarkers of this process, and might be potential therapeutic targets for these processes. So we used a number of multi-species preclinical models of inflammatory lung injury, where we exposed rats, mice, dogs, as well as human cells to stretch. And they were exposed to mechanical stress from the ventilator, as well as to lipopolysaccharide as induce a bacterial pneumonia and associated ARDS-like preclinical phenotype. We took tissue from these animals and isolated RNA, performed a number of gene expression profiling studies, and used those results to identify differentially expressed genes that might be particularly relevant to this process. And then we could select those genes for using in genetically engineered animals to see whether they in fact contribute to the ARDS ventilator-induced lung injury phenotype. As an example, here's a canine study we did with Brett Simon at Johns Hopkins, where we took a dog and lavaged one lobe, one lung in this dog, lavaged it free of surfactant. So this was an injured lung, and we left the other lung unlavaged, and so as a control lung. And we put an endotracheal tube into each lung and ventilated them differentially, so that this lung had ventilator-induced lung injury and this lung did not. As you can see, the injury is very prominent in the CT scan that I'm showing you here, both in the apex and the base. And the red circles relate to areas of the lung where we obtained tissue, isolated RNA, and then performed the gene expression studies that I shared with you earlier. So here's a heat map. Y'all probably very familiar with looking at these. In these heat maps, every column is a different tissue. Every row is a different gene. And in green is a low expression, red is a high gene expression. And you can see in the base of the injured lung has marked increases in expression compared to the base of the control lung. And overall, it's the injured lung that has higher gene expression. And it was studies like this that led us to focus on one particular gene that I'll talk about. And that gene is called NAMPT. And we'll talk more about NAMPT, what NAMPT stands for, or what it does. But the reason we selected NAMPT, you can see there's a lot of differentially expressed genes with very high expression here. And the reason we selected NAMPT was in fact for two reasons. One is that in 50 experiments that we did such as these in mice, in rats, and in dogs, NAMPT was one of the top 3 genes in almost every experiment. And so it's almost such high increases in expression in response to the ventilator-induced stress that it was hard to ignore. The other feature that led us to decide to focus and to go full court press on interrogating this gene came from studies that I'm showing you on this slide. So we took the gene expression results from those 70 microarrays that we had performed using the Pubmatrix arrays at the time. And so we filtered, and so we're comparing mechanically stressed tissues or cells to non-mechanically stressed tissues or cells. And when we looked at the genes, and we found a number of genes differentially expressed, and then we filtered them on the basis of significant gene expression, and on the basis of the fact that in every experiment, the direction of that gene expression was in the same direction, either up or down. And then when we did that, we had about 70 genes that survived this very rigorous filtering. And if you looked among the 70, you found 2 genes that were very, very common. Everyone today and even back then would recognize that IL-1 beta and IL-6 are very pro-inflammatory genes. So they're not terribly too surprising. And the way we validated that was using a informatics tool called Pubmatrix. So with Pubmatrix, you plug in a gene name, and then that allows you to blast all of PubMed for the number of papers associated with that gene and whatever headers you come up with to cross validate it. So we used the following headers, which were lung injury, mechanical ventilation, endothelium, and pulmonary endothelium. And you can see both IL-1 beta and IL-6 have plenty of papers that are associated with those particular terms, including mechanical ventilation. But what we felt was much more interesting, out of these 70 genes that we identified, we also identified 5 other genes that had very strong gene expression differential in this model. And when we blasted those genes against Pubmatrix term, we found that for these 5 genes, there were absolutely no papers linking them to lung injury or mechanical ventilation. So we thought this was a good pool of genes by which to pursue potentially novel genes that contribute to acute lung injury. And in fact, for every gene that was shown on this slide, we've published at least 2 or 3 papers validating the fact that these genes are in fact associated with ARDS severity, the risk of ARDS. And many times we've found the genetic variants in the genes that also drive linkage to ARDS. But the gene that I'm going to talk about today, and the one that's most dramatic here, is in fact NAMPT, right? You can see that there's no papers in any of these categories associated with the differentially expressed NAMPT. So when we identified NAMPT as a potential target, we did our PubMed search. And one of the reasons there were so few papers or no papers in that Pubmatrix analysis is that there was only 8 PubMed papers in the entire literature at the time. Half of these papers were in the immunology literature, talking about this gene as a maturation factor for B lymphocytes. But the other 4 papers are very interesting because they were in the OB-GYN literature. And it turns out that this is a gene that was present in amniotic membranes, and then during gestation, its expression increased. So it's suggesting that it's a stretch-induced gene. And that was really the tipping point. We decided to go after the gene. And we identified, subsequently, it was identified that this gene also functions as an intracellular enzyme. The gene NAMPT is nicotinamide phosphoribosyl transferase. So it's an intracellular enzyme that regulates NAD metabolism, the converting nicotinamide to NMN, and then through the salvage pathway generates NAD. And importantly, though, when this protein is secreted from cells, our group identified that eNAMPT, extracellular NAMPT, functions as a damage-associated molecular pattern protein, or a DAMP. The DAMPs, as you all know, are proteins that tend to bind pathogen recognition receptors. They are elaborated in response to stimuli, injurious stimuli, apoptosis, cell death, stimuli that affect the health of the organism. And what we found was that eNAMPT functions as a DAMP and binds to toll-like receptor 4, and in doing so activates an NF-kappa B dependent inflammatory pathway. And this is a work that we published a few years ago. We wanted to know where in the lung, in this ventilator-induced lung injury model, where is NAMP coming from? And what we identified is that it's primarily from activated lung epithelium. Lung endothelium is shown here on the left. Green here is NAMPT expression in a high tidal volume ventilated dog. You can see it's very prominent compared to the spontaneously breathing dog. And it's also elaborated by leukocytes in the lung, as you can see on the right panel here, mouse leukocytes that came out from bronchoalveolar lavage. The increased brown staining is consistent with NAMPT expression. So to put the mechanism of action for eNAMPT in ventilator-induced lung injury, I want to show you this slide. We've done a lot of work on understanding how NAMPT is transcribed, the factors involved in NAMPT transcription. We've shown, for example, that hypoxia is a very prominent cause of NAMPT transcription driven by HIF2 alpha. We've also shown that mechanical stress, primarily through STAT3, STAT5 transcription factors, drive NAMPT expression. And so this information is consistent with eNAMPT as a DAMP in driving inflammation. And it does so, as I've mentioned already, that it does so by binding the TLR4, which is a pathogen recognition receptor. And when it binds to the dimerized TLR4, it activates NF-kappa B and then drives a whole cascade of cytokines that enter the bloodstream, and in doing so, not only further inflame and damage the lung and increase permeability of the lung, but they also inflame other organs, and this leads to the multi-organ failure that I mentioned is what patients with ARDS die from. Patients with ARDS don't die from respiratory failure, they die from multi-organ failure. And so our strategy here would be, so if this is not only a biomarker of inflammation, but a therapeutic target, we posited that neutralizing eNAMPT using a polyclonal antibody or perhaps a humanized monoclonal antibody might have the effect of attenuating the cytokine storm, reducing multi-organ failure, and then increasing ARDS survival, which is the challenge and unmet need today. So interestingly, we also conducted some parallel studies in parallel where we obtained peripheral blood mononuclear cells from ARDS subjects. So we collected on day 0 or day 1. And when we would obtain these PBMCs, and when we compared RNA-seq data, where we now have the transcriptome for these ARDS patients, compared surviving ARDS patients to non-surviving ARDS patients, total of 55 patients in this study. As you can see, we identified a number of upregulated and downregulated differentially expressed genes. And when we plug those genes into a pathway that is linked to survival, what we found is that the toll-like receptor signaling pathway is a pathway that's very much associated with this differential gene expression in ARDS survivors. So this is consistent. Obviously, it's not direct and a linkage to NAMPT or TLR4, but it does highlight the fact that inflammatory pathways, including toll-like receptor signaling pathways, are contributing to the outcomes in ARDS patients. So how does eNAMPT activate TLR4? That's actually a very interesting mechanism there that we're still working on, and I don't want to spend a ton of time on that, but I want you to just recall that TLR4 is a fairly promiscuous receptor as a pathogen recognition receptor. It binds to LPS, lipopolysaccharide. It binds to HMGB1, another cytozyme. It binds to a number of molecules. But what's important, I think, is that each of these other activators of toll-like receptor 4 require a chaperone protein. For example, LPS requires a protein called MD2, which LPS will bind to MD2, and that will allow full engagement of the toll-like receptor 4 pathway, of the toll-like receptor 4 receptor, and engage it in the way that's shown here on the slide. Unlike LPS, however, eNAMPT does not require MD2. We have clearly shown that the activation of toll-like receptor 4 does not involve any other chaperone proteins. And we think one mechanism, a major mechanism, is the fact that the sequence of eNAMPT contains MD2-like sequences. So if we use computational modeling to look at the similarities between MD2 and NAMPT, you can see here that the loop region in NAMPT and the loop region in MD2 look very, very similar. You know, if we look at the sequence homology between the NAMPT loop region and the MD2 loop region, you can see that there's two key areas. These are the areas where MD2 binds to toll-like receptor 4. That's well known. And NAMPT contains high homology in these same areas. So we think that when NAMPT interacts with toll-like receptor 4, it contains the regions, the LPS binding region shown here in red and the MD2-like binding region shown in brown, as a mechanism of engaging and activating the toll-like receptor 4. So it's the only protein we're aware of that normally can bind to and activate TLR4 without requiring a chaperone. So what are the mechanisms underlying NAMPT transcription? Well, again, just summarize a lot of work we've done here. We have demonstrated that in response to mechanical stress, hypoxia, other inflammatory stimuli, including cytokines and growth factors like VEGF, a number of key transcription factors are activated. They're shown here on the slide. NRF2, ROS is a very prominent stimulus for NAMPT expression, and through ROS generation and NRF2 activation drives expression of NAMPT through antioxidant response elements. HIF2 alpha in response to hypoxia is the main HIF that drives NAMPT expression, et cetera. And so we've published this work, and we've also shown that the regulation of NAMPT not only involves these transcription factors, but is also epigenetically regulated with DNA methylation sites we've identified in the NAMPT promoter, as well as microRNAs and microRNA sites in the 3' UTR of the gene. And we published on both of those. And the summary of these events is that NAMPT is transcribed, it forms as an intracellular protein, it dimerizes, and it's secreted as extracellular NAMPT. So again, identifying NAMPT as a potential therapeutic target led us to generate a polyclonal antibody against NAMPT, an eNAMPT neutralizing polyclonal antibody. And we've published this in a ventilator-induced murine model in 2008. And more recently, we've used this in a rat blast trauma model where we take a rat in collaboration with folks at Fort Sam Houston in San Antonio. We expose it in a shock tube to a blast delivered to the entire rat. And then we take that rat that's just sustained that blast and put it on a ventilator for 4 hours. And you can see that there's a tremendous amount of injury that occurs in these rats that are exposed to both the blast and to the ventilator-induced lung injury, as opposed to the normal rat lung that you see here in the inset. This is a markedly, severely damaged lung where basically the alveolar space is full of inflammatory material and edema, and there's very little airspace here for movement of oxygen into the bloodstream. So we take a similarly exposed rat, but this time treat that rat with a polyclonal antibody after the blast, prior to mechanical ventilation. You can see there's a significant attenuation in the injury here, and we can summarize the injury using a severity score, a lung injury severity score, and you can see our polyclonal antibody was very effective in reducing lung injury in this model. So this led us to seek to develop a therapeutic entity that could be helpful in people. And so with the help of an STTR grant I wrote that was funded by the NIH, which allowed me to develop murine monoclonal antibodies. You can see here we screened a lot of mice for, we exposed them to the antigen of human NAMPed, and then screened them for the development of neutralizing antibodies. And after a few years, actually, we identified these two mice that hybridomas were producing producing antibodies that were very effective in reducing lung injury in our preclinical models. So with a company called Fusion Antibody, we humanized these monoclonals and we screened them. We identified the best in show for each of the 1093 and the 1076. We went through another round where we optimized the CDR sequence there to reduce immunogenicity. And at the end, we had 2 antibodies, one from each line, ALT-100 and ALT-200. So we did that about a year ago, and now we engaged Fusion to make us a stable cell line of the ALT-100 antibody, and we've gone through this stages of stable transfection, mini pools, etc. We generated a research cell bank just this past week, and now we're ready to hand this off to our manufacturer, which is a company called CMAB in Suzhou, China, and they're going to make us our cGMP antibody. So I think, given that we've already started our IND enabling studies to assess pharmacodynamic and pharmacokinetic properties as well as toxicity, when we have a batch from CMAB and can complete our large animal tox studies, I think we'll be ready to get our IND, which we think will be within a year. So that's where we are with development of the antibody, and I'm very excited about it. And it couldn't happen fast enough for me, given the fact that the landscape for ARDS therapeutics is still pretty grim. Well, we've used the antibody that we have generated, a transiently transfected humanized monoclonal antibody. I'll call it ALT-100 because that's what it is. We've used this in mouse models, and we published this last month in European Respiratory Journal, showing you that the antibody significantly attenuated the injury in this mouse model. And again, summarized here on the far right, showing you that compared to the polyclonal antibody, the humanized monoclonal antibody was more effective. And we also, in the same paper, we showed that endothelial cell-derived NAMPed is very important, because we created this endothelial cell-specific conditional knockout that cannot produce NAMPed from endothelium. And to our, we were very surprised to see how strong the protection was when you have an endothelial cell knockout. So this tells us that a large amount of NAMPed that is in the blood in ventilator-induced and LPS challenged mice is actually derived from the vascular endothelium. But I think our most compelling data for the utility of this antibody in humans comes from our studies where we created a porcine ARDS, ventilator-induced lung injury model. So we take a mini pig, shown here, instrument it, art lines, venous catheters, and monitor it very carefully on mechanical ventilation. And when we treat this pig with intravenous lipopolysaccharide and to induce septic shock while giving it high tidal volume ventilation, the results is that you have a very severe lung injury. And we can do this for 12 hours. So we keep this porcine model going for 12 hours, which is a big advantage over both mice and rat preclinical models. So again, compared to the normal pig lung, you can see here there's a lot of injury here in the septic shock ventilator-induced lung injury model. Now, then we compared these results to pigs that received intravenous ENAMPed antibody, or ALT 100, but delivered 2 hours after the onset of injury. And this is what the lung histology looks like. Pretty dramatic. It's the most dramatic I've seen in the studies that I've done with in all three species. And this is 2 hours after the onset of LPS and mechanical ventilation. And if we look at some of the parameters of injury, we see that there are ENAMPed antibody reduces levels of ENAMPed IL-6, angiopoietin-2, and IL-1 RA. And interestingly, it also reduced serum lactate levels, but did not have an effect over the first 8 hours of this injury model. But in the last 3 hours, you see that the lactate levels started to continue to had a significant bump up compared to the first 8 hours, and this was abolished by our monoclonal antibody. We've more recently conducted studies to see whether we can give this antibody 6 hours after the onset of injury. And as you can see here, we still have a significant protective effect of the antibody on BAL indices such as protein and neutrophils. And again, a severity score, which shows that at 6 hours, protection isn't as good as 2 hours, but it is still significant. So, and then finally, we conducted some biochemical studies to determine whether the antibody was affecting the pathways we thought it would be vis-a-vis the TLR4 signaling pathways inducing inflammatory injury. So our first studies were to compare, in this pig model, was to compare pigs that were treated and untreated with our antibody that were exposed to the 12-hour LPS ventilator-induced lung injury model. And what you can see here, when we looked at the NF-kappa B pathway, you can see that levels of phospho NF-kappa B compared to controls are markedly increased. And you can see that those are reduced back to controls or nearly controls by treatment with our antibody, and captured in the ratio of phospho to non-phospho NF-kappa B to the right here. Another pathway that we looked at was the AKT pathway, the PI3 kinase AKT pathway. A substantial amount of literature and preclinical models of mice and rats have looked at this pathway. And what I'm going to show you here is the pig model, again, showing you that levels of AKT dramatically are reduced in this model. And this is consistent with what we found the same thing in rats, we found the same thing in mice. And the levels of phosphorylated AKT, not surprisingly, are also reduced. And we also saw a very strong signal for increases in nitrated AKT at the tyrosine 350. We have a specific antibody that detects nitration at that site, and that is quite prominent in pigs exposed for 12 hours to this model. And as you can see on the bar graphs here, each of these AKT dysregulated effects are rectified by treatment with a monoclonal antibody. And then the final data here I want to share with you is data where we looked at mTOR and Richter. Again, like AKT, mTOR and Richter have marked reduction in the levels of protein detected. And again, this is rectified by treatment with the monoclonal antibody. And this is, so we've highlighted AKT and the mTOR pathway, at least in the biochemical studies of dysregulated pathways that we've looked at in the pig and rats here. And actually, this does link nicely to data that we've generated where we've conducted a Illumina 850 EPIC methylation array using PBMCs from ARDS patients, like I shared with you before. This time we're looking at differentially methylated sites, CPG sites, between survivors and non-survivors. And of the top 15 pathways that we identified that are differentially methylated between survivors and non-survivors, you can see here that there's a very prominent representation of AKT and mTOR signaling, again suggesting that this is a viable pathway that links to inflammatory pathways with an antibody that that might be able to be useful in rectifying these dysregulated pathways. And the final experiment before I move on to our what kind of clinical trial we'd like to be conducting here is that we took PBMCs from patients with ARDS, incubated them for 6 hours, and compared them to PBMCs from controls. As you can see here, when you look at RNA-seq data in the pathways that are differentially expressed, genes that are differentially expressed between controls and ARDS, you can see at baseline, there's a strong TLR signaling and NF-kappa B signaling signal there. Now we took PBMCs from ARDS patients and treated one aliquot with PBS and the other aliquot with our ALT-100 antibody for 6 hours. And we were very interested to note that that among the differentially expressed genes affected by our antibody are again NF-kappa B and toll-like receptor 4 relevant genes. So we think that our antibody is working through a mechanism that involves NAMPT, obviously, and involves attenuation of TLR4 firing and subsequent signal transduction events. So how do we translate this information that we have, and how do we take this asset that we believe we have and take it to a level where it potentially can improve the health of individuals that are critically ill and have ARDS from whatever cause? Well, you know, this really captures the landscape for sepsis and ARDS, in which, you know, the trials, the effect of a variety of treatments here has been very, very challenging. We've had failed trials with Xigris, failed trials with gamma interferon, failed trials with interleukin-6, more recently during the COVID pandemic. And there's multiple factors for this. Heterogeneity of ARDS is certainly one. The fact that preclinical studies don't appear to predict successful phase 2 studies. The fact that phase 2 studies that are successful are so often not reproduced in phase 3 studies, as was the case in Xigris and in gamma interferon from the company called Ferron. And an important barrier is the lack of biomarkers in ARDS that are reliable and predictive. And the fact that we know when we do ARDS trials, we don't really use any strategies to identify patients that are most likely to respond to a therapy. And so, you know, Christian Beamy is a very talented physician scientist that works with me for the past few years. He and I wrote this review article identifying this crippling gap between biomarker discovery, which is easy, and biomarker utility, which is not easy. So we've tried to address this, you know, obviously with ENAMPT plasma levels as being a key determinant for us. And I'm showing you here is some published data that where we've shown that ENAMPT levels in blood from ARDS patients and trauma patients is increased, and it's increased in COVID-19 infected patients as well. And it's much higher in hospitalized COVID patients compared to non-hospitalized COVID patients. We've also used a multiplex ELISA strategy here to measure MIF, a number of cytokines. We actually did a study with 12 different cytokines, and what we found was that these 4 cytokines, at least in our initial study that we published in 2020, MIF, IL-8, IL-6, and NAMPT, these four were able to determine with a high predictive value, those individuals likely to not survive their ARDS. So this is we're going to validate this in a very large ARDS population. We're in the middle of that right now, but this is a strategy that we think might lead us to developing a better biomarker for ARDS. And ENAMP levels, are higher in as I've shown you in ARDS patients versus controls. It's significantly related to Apache 2 scores, so therefore it's related to mortality. The linkage isn't extremely strong, and we hope to get more data like this as we get a larger sample population. And finally, another strategy that we are strongly contemplating for use in a clinical trial is to use genetic variants. So we've sequenced the NAMP gene. We found a number of SNPs, including the 5 that are shown here. And we've comprised a genotyping assay with these 5 SNPs, because these are 5 we've shown confer the risk of ARDS mortality, severity and mortality in both African and European descent subjects. You can see this. So here's the promoter. You can see where the 5 SNPs are. You can see that this is the mechanical stress inducible region we've already identified. Several fall into CPG islands. And we think that this assay might be helpful in, again, assessing risk and identifying patients most likely to respond to our antibody therapy. And if we look at these SNPs and look at the linkage between these SNPs and ENAMP levels, you can see they help drive the level of ENAMP in the blood, particularly in African Americans, and also help confer an element of risk for survival. So right now, what we're thinking about, and we've proposed to the FDA as a strategy, is to take individuals coming into the ER with impending respiratory failure from these causes, screen them with our antibodies, screen them with our genotyping study, identify people that are at risk for delivery of the antibody to them at the time of intubation for acute respiratory failure. We think this would be treating and testing a drug in patients most likely to respond to the drug. And by delivering it at the time of the ventilator, we think that we can obviate the development of ventilator-induced lung injury, decrease multi-organ failure, and probably So the last few slides, I just want to say that the COVID-19 ARDS has really highlighted a number of sequelae post ARDS. We've known for very many years that people that survive the ARDS have restrictive lung disease that tends to get better over time, but doesn't always in everyone. But survivors of COVID-19 ARDS definitely have a higher propensity of developing fibrotic lung disease. And again, as I'm showing you here on the bottom of the slide, this is not surprising, because toll-like receptor signaling, which is induced in ARDS, is also a driver of fibrotic indices. And for those reasons, and because we're targeting a ligand of the toll-like receptor 4 that binds to a receptor that is primarily driving innate immunity, it's not surprising that we would find our antibody may have efficacy in other inflammatory conditions. And so what I'm showing you here is a list of inflammatory disorders that we have generated preclinical data on. The red asterisks there means that we've received NIH grants to support development of ALT's targeting of DAMPed through the STTR mechanisms. But I just want to bring your attention to the fact that if you look at this list, quite a number of them are related to the development of organ fibrosis, whether it's radiation-induced lung fibrosis, non-radiation-induced fibrosis, liver fibrosis, and renal fibrosis. So we think that this strategy may have efficacy in long-term treatment of patients with fibrosis. And just to show you one example of that, and then I'll stop, is that we've taken a whole lung thoracic irradiation model where in this particular panel, we're showing tonsillar tissues from humans exposed to 8 gray for 24 hours. And The red here is NAMPT expression. So you can see non-radiated versus radiated. There's a tremendous amount of NAMPT expression. And we've taken mice where we expose them to a single lung dose of 20 gray, and we follow them for 18 weeks. And what you can see in mice that receive weekly antibody intraperitoneally, that weekly treatment with our antibody drops the number of inflammatory cells significantly at the end of 18 weeks, and also reduces the amount of smooth muscle actin, which is an indicator of fibrosis in the treated animals as well. And if we look at trichrome staining, again, another reflection of collagen deposition in the lung, you can see there's much less collagen in antibody-treated animals. So with that, I'm going to stop right there, and I'll just say that we're very excited about this. I think if one thing that comes out of this is that this demonstrates, I think, at one level, power of genomics. You know, we identified a gene through what's often disdainfully referred to as shotgun exploration, but we identified a gene that was not present in the literature in any way, shape, or form related to lung disease. We found that gene actually encodes a protein that serves as a biomarker for lung disease. We've identified that protein as a target for therapeutic intervention, and that polymorphisms in that gene confer increased risk of inflammatory injury and also increased severity of inflammatory disease. And so hopefully through the talk I've just given you, it demonstrates the fact that we were able to leverage these system biology approaches here that I think have led us to a point here where we think we're on the cusp of being able to maybe deliver some precision ICU medicine. And Lord knows that we can't wait any longer. Our field and our patients definitely need this. So I'm going to stop right there and say thank you very much to Linda again for the warm introduction and the invite to be here, and I'm happy to answer some questions.
Thank you so much, Skip. This wonderful talk. It's amazing the recent progress in NAMP, research and in the peaks and all that development, the stable cell line. That's just wonderful. It's instantly will be into the clinical practice, hopefully. So we have some questions coming up.
Okay.
The first is from John Belperio. His question is, do you say the neutralizing antibody primarily reducing inflammation, or it also affects permeability?
Yes, that's a great question, John, and I'm glad you're on the call to see that my Philadelphia Examiner picture there. So the antibody doesn't directly affect permeability per se, but the ENAMPT, by binding toll-like receptor 4, that's a permeability inducing signaling event. So if you can block TLR4 activation, whether it's LPS or whether it's ENAMPT, you will reduce vascular permeability, and we've shown that. So that a reduction in vascular permeability is, in fact, I think, a very, very important point that you're making. It's an important outcome of dampening TLR4 activation.
Okay, the next question is from Stephen Chang. Nice talk. I may have missed it, but I'm guessing that the LPS villi peaks were ventilated with large tidal volumes. Does ENAMP neutralization work in non-villi ARDS, for example, LPS only, since
Yes.
it is standard of care to ventilate with 6 cc per kilogram IBW? And yeah, sorry, please.
Yeah. So that's a great question, and we have shown very much that in just a pure LPS model, whether it's an LPS pneumonia, whether it's a cecal ligation sepsis model, we have demonstrated that the antibody itself works very, very nicely in attenuating injury. And the reason we think that is that in the LPS model, ENAMPT is prominently induced to be secreted. So ENAMPT's part of that cytokine storm released by the LPS and neutralizing that has definitely a beneficial effect. And we've also looked at that blast model that I showed you. We've looked at rats exposed to the blast model without ventilator-induced lung injury, and the polyclonal antibody in those studies was protective as well. So yeah, we think that this has utility beyond ventilator-induced lung injury for sure, and I think that's part of Stephen's point. But of course, you want to get your drug approved by the FDA. We're trying to highly focus the clinical trial design to have the best chance of knocking out ventilator-induced lung injury, and that's kind of the strategy we're using right now.
Yeah, he has another line. What I wonder is if neutralization of ENAMP could potentially help in a situation in which patients have been properly managed with regards to lung protective ventilation.
Yeah, that's a great question too. And what I will answer to you for that one is that if you look at levels of inflammatory cytokines in patients that even have the most rigorous utilization of protective ventilation, you still see inflammatory cytokines in their blood. They're not as high as when you're not using protective low tidal volume ventilation, but they're up. And I think that's an important part of the equation here, is that anything you can do to further reduce the burden, the inflammatory burden here, is important. And the final point is that normally cytokine levels would drop for a number of different inflammatory cytokines, but because NAMPT is very much driven by mechanical stress is a major driver of NAMPT expression. So if you are still on a vent, you're still driving NAMPT expression, even if it's a low tidal volume. That's not a normal breath. Your lungs are still very heterogeneously affected. You're still producing NAMPT in the blood, and we've shown that by measuring NAMPT in people over time. But it's a really good point.
Okay, so next question from Marlene Thomas. Since NAMPT is expressed, it's jumping, in the amniotic fluid from fetal lung epithelium, any potential role and application in ARDS of prematurity and chronic lung disease?
Oh my gosh, thank you for that. I could talk for 20 minutes about this. This is a great point. So I don't know if you noticed on that slide, I had all the indications. One of them was for chorioamnionitis, right? So we are we have a lot of data on this. We've shown that mothers with chorio, which means intrauterine infection, okay? Pregnant mothers with intrauterine infection have higher levels of NAMPT in their blood. Pregnancy also increases NAMPT in your blood, so that's not such a great biomarker. But there's several papers that have shown measured amniotic fluid NAMPT, and amniotic fluid NAMPT is very high in people in pregnant moms with chorio. And those levels are very linked to premature delivery and the untoward outcomes of having severe premature delivery of infants. So that's an important point. So we think it's a viable, and we've looked at the placentas of women with chorio, and their placentas light up with NAMPT expression. Now we've also done a murine model of chorio when we inject LPS into the pregnant mom to induce chorio. They deliver their pups 48 hours later, 24 to 48 hours later. 80% of those pups are dead. And if we treat that animal, treat it with LPS, but then give it the antibody within 12 hours, it goes from 80% mortality to 80% survival. So we really believe, and we just got a grant from the NIH and Child Development Institute to evaluate this antibody as a potential therapeutic for pregnant women at risk for premature delivery. Because once you have inflammation and you're pregnant and you start to have inflammation in your amniotic fluid, the placenta will start to break away, and that's the signal that's the signal for inducing delivery. And so that's permeability. That increases permeability in the amniotic membranes. Long-winded answer, but great question. Thanks.
So Stephen has another, seems like the irradiation model of lung injury argues for potential of ENaP neutralization in ARDS.
Yes. If he's asking, does our data in ARDS similar protection in radiation-induced lung injury? The answer is yes.
Okay. And the next one from Thomas Graver, can you elaborate on why prostate cancer made the clinical trial list?
Well, this is great. I'm glad. Thank you so much for that question. So here's the thing. We're interested in prostate cancer. We have similar data with lung cancer, but I think the oncology folks know that the interface between inflammation and neoplasia, they're intimately linked. Inflammation is well known to be driving the development of aggressive cancer behavior. And we think that that's very prominently a part of prostate cancer pathology. So we published a paper last month in eBiomedicine where we evaluated men with prostate cancers ENaP levels. If you had a low-grade prostate cancer, the levels were low. If you had a metastatic profile, the numbers were high. We looked at tissues, prostate tissues, from normal prostates, from prostate gland confined cancer. So you had a positive biopsy for cancer, but it's confined to the prostate, and compare that to prostate cancer that's escaping the gland. And you'll find that NAMT expression goes exactly along those three lines. Almost no expression in normal tissues, very mild expression in gland confined tissues, and very extremely, I could show you some slides of that, extremely prominent expression in invasive cancer. And the mechanism behind that, we think, is that there's a periprostatic fat that's exposed to hypoxic environments. And we think that elaborates a lot of HIF2 alpha, which drives NAMT expression. And these inflammatory cytokines in the tumor microenvironment, we think, convert an indolent cancer to an aggressive cancer. We've injected our antibody into SCID mice that we've already previously injected human prostate cancer cells, and our antibody blocks the invasiveness of those prostate cancers into the diaphragm. So it's all in that paper, eBiomedicine. Belinda Soon is the first author. But we're very excited about these data, actually, and we've shown the same in intraprostatic injected cancer cells, that it's decreasing prostatic volume and increasing survival in those animals.
Okay, so the next from Ari Srivasan, whether all 5 SNPs are required as a biomarker or a single or 2 SNPs are sufficient as a clinical marker to identify the number of SNPs to ENaP expression and survival?
Yeah, great. That's a really, really good question. 2 of those SNPs are pretty much in LD, but not exactly. So we kept them both in. You don't need all 5 SNPs to have high risk. The 2 SNPs I'm talking about alone definitely increase risk, and more so when they're both present. But through the 5 SNPs, we cover African American polymorphisms as well as European descent polymorphisms, and so the 5 SNPs we think is a very, very good attraction to identifying who's at risk for the inflammatory disease that we're talking about. ARDS could be radiation-induced lung injury. It could very well be prostate cancer, but yeah, you don't need all 5 SNPs.
Okay, wonderful. We addressed all the questions.
Hey.
Yeah. Okay, so if no more questions, thank you so much, Skip, again, for joining us today. It's a truly wonderful talk. We learned so much.
Linda, you're very, very kind. And again, I'm looking forward to talking to you later on this afternoon. And, my thanks to everyone on the call for your attention and hanging in there, and also for your great questions. So take care and stay safe, okay?
Okay. We're signing up for this. Yeah.